All right. All right. But did anybody remember any questions that they had from today's exam that they wanted to ask, or if not, that's fine. Cool. All right, we're just going to do chapter 16 today.
Sweet, I love this stuff.
Not that difficult. It's really not that difficult. A lot of it we've seen before. Structure, composition, and function of nucleic acids, DNAs, and RNAs. Oh, shoot, the batteries. All right, got them replaced. All right, so nucleic acids, what are they for? What do we use them for? DNA, RNA, most specifically, yes. What we need to know what composes them. So there's that little formula. There's always the three things. They're composed of a nitrogenous base, a ribose sugar, or a phosphorus group. Got to know those three things. So whenever you see nucleic acid, whether it's a DNA or RNA, it's going to have the nitrogenous base. That's going to be your purine or pyrimidine, your adenine, guanine, cytosine, or thymine, yes? Or maybe even uracil for RNA.
Did you say or phosphor?
And. That'll be an and. It's going to have your ribose sugar, so either your deoxyribose or ribose. Deoxyribose is probably going to be for DNA. Your regular ribose is just going to be for RNA. Pretty simple, right? That's how we got their names. And a phosphoryl group. Excuse me. It's really, we typically associate nucleic acids with our DNAs, RNAs, what have you. But the rest of this paragraph, or the next paragraph, pretty much says it's actually in a lot more things. It's in our ATP, right? So adenine or adenosine, whatever it is, or GTP, guanine. So in all of our high energy molecules that we have, we see our nitrogenous bases. Our cyclic AMP or cyclic GMP, those second messengers, has adenine in it too, or guanine. All of our electron carriers that we learned in the electron transport chain and for this previous test, NADH, NADPH, FAD, all those have those nitrogenous bases in them as well. So very important. So probably we need to make sure that our diet and our nutrition has adequate enough of this, right? So that we can actually incorporate these into all the inner workings of our body. Pyrimidines, let's see, where are we at? Are also utilized in high energy, something else, whatever, we're going to go on. There's two basic kinds, DNA, RNA. DNA is considered the repository for genetic information of cells, whereas RNA is the expression of DNA. So our information is stored in DNA, and RNA is where we kind of take it out of storage and we use it. We make proteins or enzymes or whatever else needs to be happen. Reproduce a cell, replicate the cell. Genetic information stored in DNA must be stored in a stable form, right? So if we, when we put stuff into storage, we'd rather have that temperature, climate-controlled one as opposed to the one that's like a shack and is like boarded up. That's not as good storage. It's not as safe. So we want to try to have our DNA as safe as possible, our genetic information as safe as possible, and is decoded by transcription, translation. We'll go down there here in a second. And those two things, transcription, translation, is what converts the nucleic acid sequences into protein sequences. So going down here at the bottom, need to know the differences between transcription and translation. We talked about this in test 1, I believe. Do we still remember that? Yes. If not, DNA goes through replication, making another strand of DNA. It replicates itself.
So we have transcription, translation, and then transamination.
Transamination is way different. So transcription is going from DNA to RNA. We are oh shoot, how did I do this? Like transcribing it. Like scribe is to write it, is to write. So we're kind of taking it and writing it a different way, that information. And then translation is going from RNA to protein. Transamination is when you are taking an amino group from one molecule and putting it onto a different molecule. So that's just the amino, like the NH3 specifically. Does that answer your question? All right, so we need to know the difference between replication, transcription, and translation. I think we've covered that now, and Dr. Ash should have covered that as well. Replication is where DNAs make more DNAs. Transcription is when DNA goes to RNA. Translation is when messenger RNA makes proteins. Oh, that's not the button I want. All right, chemical components. So these are the three things that we talked about earlier. Gotta know them. A nitrogenous base, deoxyribose sugar. This is for DNA, right? So if we see deoxyribose, we're thinking DNA, we're not thinking RNA. And the phosphate group. And there's a nice little picture of what that all looks like. And our DNA is just a bunch of those stacked next to each other over and over and over again. Nitrogenous bases. We've got two types, purines, pyrimidines. Our purines are adenine and guanine, and our pyrimidines are cytosine, thymine, and uracil. How I remember that, I like to do put those three and make the acronym CUT. So those all go together, making a quick little three-letter word. I liked word searches when I was younger. Still, I guess I still kind of do. Anyway, here's what they look like. Our pyrimidines are, if these were all just carbons and hydrogens, what would that be called? Going back to like way chapter 1 or 2. What would this be called? Potentially. Six-member ring. Yeah, like a benzene ring, right? And if we wanted to attach that somewhere, that'd be like a phenyl group. Yes? All right, so our pyrimidines kind of look like benzene. Pyrimidine, benzene. That doesn't really work, purine. And our purines pretty much have this extra kind of pentamer next to it. He's not going to ask you about what the structure or anything, but that's what it is. Purines are A and G, and cytosine, thymine, uracil are the pyrimidines. They're all related. We'll get that here in a second. But moving on, we've got nucleosides, nucleotides, and nucleic acids. They're all a little bit different, and he might ask you the difference on the final exam. So a nucleoside is composed of a purine or a pyrimidine base covalently linked with a pentose sugar. So going back over here, it's just the base with the sugar. The nucleoside does not have the phosphate group on it. The nucleotide is that nucleoside plus the phosphate group. So really, nucleotide is the three, has all three things. Nucleoside, subtract the phosphate. Does that make sense? Nucleoside, subtract the phosphate. Thus, DNA is a polymer of a lot of those big words, what have you. We've got a nice little box here at the bottom of the table that kind of lists everything. So nucleoside is just the base and the sugar. Nucleotide is the base sugar and the phosphate. And then that makes, they're either a part of DNA or RNA. So nucleic acids are going to be multiple nucleotides, right? We learned that chapter 1 or chapter 2. What makes up proteins? What amino acids are the functional group, right? And then for carbs? monosaccharides, maybe. Yeah, monosaccharides make our polysaccharides, which make our sugar. Yeah, yeah, okay. Saw a couple light bulbs. So this is just saying that nucleotides are kind of the functional unit, and they make up either DNA or RNA. And you can see here the nucleoside has an S in it, so it all ends in SINE, sine. So adenosine, deoxyadenosine, versus the nucleotide is adenylate, deoxyadenylate. They all have the T's in them. So it's really simple. If you see anything on the exam that has an S in it, in the suffix, it's probably going to be a nucleoside. And a nucleoside does not have what in it? The phosphate group. So that means it has the what in it? The pentose sugar and the base in it, right? The nitrogenous base. Good. Good. Okay, here's just more pictures.
Do you think they'll ask us to identify these?
He shouldn't ask you to identify that, no. That's not major enough. I don't believe. All right, next page. We're on page 191 here. Hopefully our books are the same. TQ here at the top. Phosphodiester linkages are the covalent backbone of DNA and RNA. That was like the big test I missed when I was an undergraduate. I saw that and I was like, wow, I don't know what that word is, so I'm not going to pick it. Y'all see this word, and you're going to know it, that the phosphodiester linkages are the covalent backbone of DNA and RNA. What makes the bonds for proteins? What types of bonds? Peptide bonds, right? This is the equivalent of peptide bonds, but for nucleic acids. Phosphodiester. So a phosphate group with two ester linkages on it. We can see, so going into the DNA, the phosphodiester really is just the phosphate group is in between the two sugars, pentose sugars. So it goes pentose sugar, phosphate group, pentose sugar, with the nitrogenous bases hanging off on the inside. Phosphodiester. Another thing, joins the 3' carbon of one sugar to the 5' carbon of the next sugar in the same chain. All right.
Okay, I've always gotten confused on this. So it goes from the 5' end to the 3'.
Yes.
But then it adds to the 3', or like.
We'll get to it. We'll get to it later. We'll get to it later. Overall structure of DNA, it's a double helix, right? We've all heard DNA is a double helix before. My grandfather was a chemistry, he actually has his PhD in chemistry, and he got that in 1951, I believe. So he was actually in school when Watson and Crick figured this out. So he was on like the cutting edge of all this microbiology and biochemistry, and so that just blew everyone's mind. So something that we kind of just like take for granted, like, oh, it's just a double helix, like whatever. They won the freaking Nobel Prize for it. So, like, kind of a big deal. Sorry. So each DNA molecule is 2 chains, and the chains are joined by hydrogen bonds. Hydrogen bonds, that's a test question. So we need to make sure that we get that straight and that the phosphodiester linkages, phosphodiester bonds, join the different nucleotides in one chain, whereas hydrogen bonds combine 2 chains together. Similar to how when we were making proteins, right? So the peptide bonds were what made the primary structure, but then the secondary and tertiary structures were hydrogen bonds together. Yes, it's the same thing. Same concept. And they pair in a really specific way. The adenine on one chain forms a base pair with the thymine on the other chain with that hydrogen bond. So A and T, adenine and thymine, are always paired together. And guanine always pairs with cytosine. If, I don't know, it's trying to think of a good example. It's like when y'all go to Palp Lab and you always pick the same partner every time, really, even though they say not to. Kind of like that. So the A and T, those two form 2 hydrogen bonds. And the C and G contain 3 hydrogen bonds. Looking down here at this page, we've got adenine and thymine. Based just how they are structurally, there's really only 2 spots for them to kind of come together. So there's 2 hydrogen bonds based on their structure. Guanine and cytosine, just how they're able to come together, they have 3. How I remember that is AT2. 2 like AT2. T and T kind of go together, and then GC3 rhymes. So GC3, AT2, or 2 and TA, just spelled wrong. It's like ta, tu, ta, atu. It's like a atu. It's like a really weird sneeze. The base sequences of the 2 strands are complementary. The chains on a typical DNA strand are anti-parallel. That's another test question. They run an anti-parallel. What the heck does that mean? That one chain runs in the 5 to 3' direction, and the other runs in the 3 to 5' direction. So, scratch paper. I'm going to use the take-home exams. Take-home exams. What were these? What take-home exams are y'all doing? Y'all had a take-home exam for D? Okay, I'm about to. Oh my God, he's slacking. All right, what the heck does anti-parallel mean? We've got anti-parallel, parallel. So anti-parallel, we've got one strand going from 5' to 3' this way, but it's going from 5' to 3' that way. So they're running in different directions. If they were parallel, they would both be running in the same direction like that. The anti-parallel makes it more stable for whatever, just for the way that the base pairs can line up. It makes it more stable.
Does anything run parallel?
I don't know the answer to that question. I'm sure that some things do. I'm not really sure, to be honest with you. RNA is single-stranded also, typically, so it doesn't really run any direction with anything. All right, anti-parallel, got to know it. DNA is anti-parallel. And it forms grooves, it forms major and minor grooves. It's not a perfect helix. It's not a perfect double helix that it makes. So a single helix would just be one, kind of like a ribbon hanging down. Double helix means there's kind of like 2 6-inch ribbon curls. 6 inches. Sorry, Elf? Nobody? I got to watch it tonight. No, maybe. All right, so these helixes aren't perfectly symmetrical all the way down. There's a major groove and a minor groove. And we learn why the heck that is up here. The double helical structure of DNA has 2 alternate period, whatever, along their long axis. One was demonstrated as having a minor groove of 0.34 nanometers, and the other one has a major groove of 3.6 nanometers. So the difference, he actually could ask you, I think I remember him maybe asking on a quiz, how many, the distance between the minor groove to the next minor groove was calculated as like 10.5 base pairs. I think I remember him pulling that out somewhere along the line. Or maybe that was just what stuck with me from learning this. But 10.5 is kind of from groove to groove. So from minor groove to minor groove. I jumped at.
I'm confused now.
Okay.
The words say 0.34 nanometers. The picture says 3.4. What is that?
Those are amperes.
Yeah.
So
Smaller than
divide it by 10. So if you can see here is like 36 amperes.
Oh.
So I think 1 ampere is like 0.1 nanometers, I think.
Okay.
So just look at the units. I skipped a little bit ahead. We need to go back to the top of the page up here. Chargaff's rules. Test question. You need to know it. Yes, so this is for base pairing. So this is DNA is double helix. In all cellular DNA, the number of adenine residues and thymine residues are equal. And the number of guanines are equal to the number of cytidines. Cytidines. I thought it was cytosine, whatever. Why would that be? Why would we would have adenines and thymines equal to each other?
You can't have odd pairs.
You can't have odd pairs, and those are what bind together, right? So if you have an adenine in a DNA, you pretty much have to have a thymine on the other side to bond to it to make it the double helix and make it stable. This is why the number of purines, which ones are our purines? A and G, and our pyrimidines, which would be C, U, and T, right? Cytosine, uracil, and thymine, are equal. So purines equals pyrimidines due to those base pairings. And that's called Chargaff's rules. So if you see on a test, what is Chargaff's rules, the answer is going to be the number of purines is equal to the number of pyrimidines. And that's due to the base pairing. It's really that simple. The DNA helix. He had something on here as well. It feels like I'm making a lot of CQs, but this is just what I remember. There's 3 different types. So there actually is, these are still, I think these are still running anti-parallel to each other, not parallel to each other, pretty sure. So the B DNA is the classic Watson and Crick, 1951, my grandpa's old. This is a right-handed and contains 10 base pairs per turn, right? So when we were doing proteins, was the right or left-handed one more stable?
Right's always more stable.
The right was more stable, yes? And so it makes sense that the right helix in DNA would also be more stable, yes? So standard, how I remember that is you need to be right. Don't be wrong, be right. Okay, because I think he asked something about which of the following is the Watson and Crick model for DNA, and he had these 3 answers. And you want to be right. You don't want to be wrong. Then we've got A. I don't know. I guess A was the pilot or the first draft, and it got nixed. So then they came up with B, and that was the right one, which is why we're using B. I haven't really figured that one out yet. But this one's a little bit wider and flatter, but it's still a right-handed turn. So A and B are right-handed, whereas Z is left-handed. And there's a nice little picture on the next page, kind of how they all compare to each other. Watson and Crick is kind of right in the middle. Huh, right in the middle, get it? Huh? No, okay, whatever. Need to know, Watson and Crick, B form, right-handed. A, right-handed, but not as good as this one. and then left-handed Z.
Eyeball
Oddball out. I'm going to let a lot of y'all just kind of, I'm going to skim a little bit of these next couple sections, and if y'all want to read more in depth to it, you can. Or I guess pay attention when he's talking about it in class, because I don't think he really emphasized a lot within these next couple pages, but I think y'all are going to have a better gauge for that when y'all are coming up. All right, structural organization of DNA, epigenetics. We're not going to be doing a lot of epigenetics. That's nice. DNA, if we put it all out next to each other and ran it out, it'd be 2 meters long, which is how many feet? How many feet is 2 meters? It's like 6 feet, right? All right. Okay. 6.
3 feet per meter.
It's about 3 feet per meter. Yeah. Yeah. No. So we've got 2 meters of DNA in our body that needs to kind of be stored in a lot less space than kind of 2 meters, right? It needs to be in every single cell. So how this happens is we use histone proteins to kind of wrap around. So when we're doing when we have our Christmas lights, we don't really just kind of do the like around the hose with it. We kind of have that. I guess if people actually have Christmas lights, there's that little plastic thing that you wrap it around to keep it all stored up nice and tight. Maybe I'm just really old and no one does that anymore. I'm only like, yeah, like there's like a spool, right? For yarn or something, you wrap it around a spool to condense it. And that's all a histone protein does. Is it's the spool for our DNA. So the genome of eukaryotes, I'll read just a little bit so y'all can just sit back a little bit. The genome for eukaryotes, including humans, is composed of multiple chromosomes of varying sizes containing DNA. The total extended length is 2 meters. However, the DNA must fit into a nucleus of a diameter of only 5 to 10 micrometers, a lot less than 2 meters. How many feet? 6 feet. Good. Chromosome is a supercoiled DNA molecule present in the nucleus of all cells. This is the first time we're seeing the word chromosomes. So all chromosomes are coiled DNA. And they're coiled around proteins called histone proteins. Fun, I guess, it's not that too important, but the proteins are basic in nature, and since they're nucleic acids, they kind of attract each other, which is why they're able to coil so tightly. Just those attractions. The basic structure of a chromosome, where DNA is wrapped around the histone proteins, is known as nucleosomes. I don't think that'll show up, but is that bolded? Yeah. All right. Yeah, the other's just kind of for your own good. And there's a cute little picture of it. So chromosomes are condensed DNA, right? So when we did like Mendelian genetics in biology, did y'all do that with cells and tissues? Like when we're trying to replicate and we have our chromosomes, but then they like uncoil and like the DNA actually like splits and divides. No? All right. Don't worry about it. We'll skip it. Gene expression. All this is saying is what turns on and what turns off our gene expression. So if we have chromosomes, tightly coiled DNA around these histone proteins, do you think that's going to be actively replicated, or do you think that's kind of being in storage mode? If it's storage mode, right? We've got our yarn around our, whatever we said it was, spool, or our lights around our little plastic thing, probably also a spool. We're not using them, right? So, but when we unwind it and we stretch it all out, we have our DNA, and that's ready to replicate, yes? Something's got to be able to tell us when it's time to replicate, when it's time to just chill out and just be there for when we need you. And all that is with acetylation and deacetylation. Just let me refresh here. I think acetylation, when the histones are covalently modified by acetylation, it relaxes or unwinds the DNA. I'll just underline it here. This is where we're at. So if the histones are acetylated, if an acetyl group is put onto it, it unwinds the DNA. It can be ready for dividing. Then if we take that acetyl group off, the histones can bind to the DNA more tightly, and it goes back into storage. That's all that big paragraph is saying. And then there's this little picture at the bottom, kind of telling you all about it. Neat. DNA methylation. Y'all can have fun with that. I'm just going to skip that. Is that cool? Cool. Reversible denaturation. Well, with proteins, what did we say could denature a protein? Acidic environments, heat, or like basic environments could as well. So just pH changes, those types of things, temperatures. So those are the same things that can denature DNA. It's the same stuff, which makes sense. So if we have too acidic of a pH, our body, what's our body's normal pH, resting pH?
7.35
to 7.45, yeah, 7.35 to 7.45. If our body becomes too alkaline and maybe we get up to 8, and our proteins aren't working, it probably makes sense that our DNA is not going to be working either, right? Because that's not in the optimal range. Everything's so sensitive to our body. That's why homeostasis is such an important thing, and why chiropractors do such a great job of maintaining homeostasis in our body, keeping everything aligned and our nervous system free to function, tying it back. Okay. Denatures causes reformation to the base pairs, so we can. The base pairs are held together by what types of bonds? The adenines and thymines, and the guanine cytosines. Hydrogen bonds, right? Hydrogen bonds, the phosphodiester bond is what is the backbone of the DNA, keeping the entire, like one strand together. When we denatured proteins, we broke the hydrogen bonds, right? They're just easily broken. They're weak individually, but together they're pretty strong. So those hydrogen bonds broke, protein denatured. We put it back in the right environment, renatured spontaneously. Yes? Innate intelligence, our body's brilliant. Yes. Same thing with DNA. The hydrogen bonds are the same bonds that are holding proteins together. These hydrogen bonds that are keeping two strands of DNA together can split and then put back into the right pH or the right environment, take away the heat. They come back together. Innate intelligence. And that's what this picture is showing. It's the same picture as proteins, but just with nucleic acids. Hybridization is when DNA can pair with RNA, it seems like. Whatever. I don't think that's that big of importance in the grand scheme of things. Replication is semi-conservative. That's a potential test question. It's semi-conservative. So it's like not really conservative, but it's kind of got some liberal ideas going with it. So each DNA strand serves as a template for the synthesis of a new and complementary strand. Thus, the parent strand synthesizes two daughter strands with complementary base pairs. What the heck does that mean? Semi-conservative. It's nice and color-coded for you. Got our parent molecule. Those two split, and it's semi-conservative, so one half of each parent cell goes with the new daughter cells, I guess. And then when those split, it's kind of the same thing. You take one half of each makes one half of the second generation. So we've gone from 1 to 4. Who knows? Does anybody know, just a tangent, what the other type of replication is besides semi-conservative? No? Is there like another type of replication besides semi-conservative? Like, does conservative just like, what does that do? I have no idea. I just picking your brains. Whatever. We tried. We tried. We're tired. It goes bidirectional. When we are replicating DNA, we go in both directions. It makes sense that we would do that just because we can save time. Yes, if we're doing both at the same time, we can do twice the work in half the amount of time. I just said time a lot. Goes in both directions, but each direction is, one direction is a little bit different than the other direction. We have something called a leading strand and a lagging strand. Have you all seen this before? Yes. All right, so I'm just going to kind of briefly touch over it. The leading strand is continuous. The lagging strand is non-continuous, and it's kind of pieced together, stitched together at the very end by these Okazaki fragments. In the leading strand, DNA synthesis proceeds by the separation of parent strands, followed by unwinding ahead of the replication fork in the 5 to 3' direction. So the leading strand is in the 5' to 3', yes? Yes, and then the lagging strand is in the, what? Is that also the 5' to 3', or does that go the other way? I think they're both 5 to 3. Right, because they're anti-parallel? Ooh. Yeah. Yeah, so both of them go 5' to 3'. I remember when I first started trying to learn these, I was always confused by which ones go which direction. So replication always goes 5' to 3' for DNA. The only thing that's different is that the DNA polymerase 1, which is responsible for unwinding all of this, goes in the 3' to, or DNA polymerase 1 catalyzes the DNA synthesis. It copies or reads the parent strand in the 3' to 5' direction. And the synthesis goes in the 5' to 3' direction. Typical DNA, we're going to read it 5 to 3. It's going to replicate 5 to 3. But the thing that's doing the replication, that's making the new strand, is reading it from 3 to 5. That's the only one that's 3 to 5. Sounds good. You get on board with that? It requires a single unpaired template strand. So this needs to be they need to be split first, and then the DNA polymerase 1 can read it going this way, and then it's going to lay it down from the 5' to 3'. It needs the deoxynucleotides, so these DATP, DGTP. Do you remember what those? Ooh, I think actually that's going to be, is that the next chapter? Ooh, that's the next chapter. It's an exciting one, actually. It actually made a lot of sense when I was learning it. And we need these. Those are what, pretty much we're going to take off the D when we put them all together. He's not really going to ask you about that. Moving on, chain elongation of the DNA chain. It goes from the 5' to 3'. The DNA polymerase 1 is reading it from the 3' to 5', because what that's doing is There's the polymerase 1 is on the template strand. Okay. What it does is it says, hey, here's a T. So the base pair with T is A. So that D, what was it? The D ATP comes in, attaches to the polymerase, and it's paired. Then it goes to the next one. We've got a C. It's reading it in this 3,5 to prime. We've got a C. All right, we need a G to pair with the C. So come in, bring the D, GTP, boom, we got a G. Now those are paired. Go, what's the next one? And so on. So that's how it's made. That's why the polymerase is reading from 3' to 5'. It's because it's actually on that strand, but the other one's being built anti-parallel to it. I think there's something else that actually unwinds it. I think the polymerase is what's actually putting on the, it's actually making the DNA strand. That's my understanding. Is there someone who agrees or disagrees with that? Is there, is there a different enzyme? What's the enzyme that unwinds the DNA in the first place? I think it's the replicating one. There's something else that unwinds it. Maybe it does both.
So which one's 5 to 3? I'm just confused.
Okay,
Sorry.
that's okay.
No. 5 to 3 is the one that's been replicated. Because it's being read 3 to 5.
Let's bring back old take-home exams. All right. We have DNA. 5', 3', 3', 5'. Yes? Okay. These need to split. Okay, so something's going to split it. So now we've got, I'll just put them vertical now. So we've got a 3' over here, 5', and then this one over here is going 5' to 3'. That's a 5. I'm working on my handwriting, not really though. Okay, good? These are two separate strands now. What DNA polymerase is going to do, DNA polymerase is going to come on here at the 3', and it's going to synthesize a new DNA strand. Okay? So this is going to go down this way from the 3' to the 5' end. And that's because this new strand being made here is being built in the 5' to the 3' direction. So that's the same thing over here. So the DNA polymerase would be on this side, on the 3' end. So it'll start building that way. Yeah, helicase is the one that unwinds it. That's right. Does that answer your question? Cool. Good old take-home exams. So that's just a picture of how it's done. DNA synthesis in the lagging strand, that was the leading strand. DNA polymerase 1 is dealing with the leading strand. In the lagging strand, the DNA synthesis, that's not centered at all. I apologize. On the lagging strand, DNA synthesis occurs via the formation of the Okazaki fragments. In the fragments, RNA primers are attached, and then eventually they remove the primers and fill in the gaps. The gap is filled in by attaching the right nucleotide based on the lagging strand, and seals the gaps by DNA ligase. So DNA ligase is, I guess, the tailor who fixes everything up at the end. And here's a nice little, DNA helicase is what unwinds the DNA in front of the replication fork. And that's I have no idea what this topoisomerase is, to be honest with you. I've never seen that before just now. So I think he blew through a little bit of this section when he taught it with us. So if he does that, don't be surprised. But if he doesn't do that, also don't be surprised. So be ready for everything. That's what I'm saying. Ha 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 ha. Yeah, there's the laughs. There it is. They're fake. It's okay. DNA mutation. Permanent damage to the DNA nucleotide sequence is known as mutation. Permanent damage, mutation. All right, like the X-Men can't not be Wolverine anymore, right? That's permanent. What? Okay. I haven't seen the new Wolverine movie, so I don't know if there's something that happens there.
It's the third Wolverine.
Oh, the X3? Yeah. Oh god, yeah, I didn't get past X2. Anyway, but there was not a cure. Well, they chose to not accept it. So they stayed mutant permanently. That's what I'm going with. These alterations are permanent changes in the base sequences. So those permanent changes are then seen in the daughter cells. And those are due to replication errors or damage by radiations, toxins, chemicals, what have you. This is how we get cancer, right? Our DNA stores our information. For people with skin cancer, we don't put sunscreen on, and those ultraviolet lights come in and cause damage to our DNA. And what that can do is that can either change a guanine to a cytosine or a guanine to a thymine. Something as simple as that can then lead to proteins not working or proteins working too much, what have you. So DNA mutations are kind of a really big deal. And it's really surprising. I think we'll get to it later, but we have 6 feet of DNA in our body. We have how many trillions of cells in our body storing this DNA. We're constantly replicating, constantly, that DNA polymerase 1 is constantly making new strands at a ridiculous speed in our bodies. But very few errors are actually made. Or the errors that are made are just silent and they don't really matter. It's kind of cool. Innate intelligence. Types of mutations, there's different types. 3 main ones, gotta know them. Point mutation is a substitution of one base for another. So that's the one I just talked about where either an adenine can become a cytosine, whatever. It just changes one letter. An insertion mutation is the addition of one or more nucleotides within a DNA sequence. So say you had A, B, C as your sequence. I guess we'll make it actually a DNA. Say we have like AAA. Now with the insertion mutation, that's going to be like A, T, T, AA. It's inserting itself in there. Deletion mutation is the removal, so it's the opposite of the insertion. So that would be going from like AAA to just 2 A's.
So then that means the daughter strand might get off track too.
Exactly. So all these types of mutations can potentially have effects on the daughter strands. DNA repair. Our body knows that we've got trillions of cells in it, and mistakes might happen. Right? We make mistakes, and we're just one person. So the body makes mistakes, and it's 10 trillion kind of people, if you think about it. But we're mostly bacteria. I heard we're like more than half our weight is actually bacteria in our body. Someone said that one time. I don't know. I never fact-checked it, so spreading slander and lies maybe. But DNA repairs. There's ways to fix it if there are problems of misreading nucleotides or what have you. So what happens is it can remove the segment of DNA that has the damaged portion or the mismatched bases, with exonuclease or endonuclease activity. I don't think you'll need to know those, so like don't really worry about those enzymes. But it can take it out, and DNA polymerase will just kind of try it again. So those come in, take out the segment, if the train track is broken, it can just come in and take that one piece out and replace it with another piece. And then ligation to the rest of it. It kind of just bandages it up together. The main enzymes that you'll need to know are the DNA polymerase and the DNA ligase. I don't think he'll ask you about the other ones, but they are bolded, so just see what he emphasizes when he lectures. We actually are ahead of schedule for the first time. I'll try, so that's exciting. That's pretty much it. Get the heck out of here. Go sleep.